Hey all, hope you all are doing well. Today we are going to discuss the important topic of lipid metabolism, that is beta oxidation of fatty acid. So before going in the detail of beta oxidation of fatty acid, please subscribe my YouTube channel, that is Biochemistry Basics by Dr. Amit, and don't forget to press the bell icon so you can get all the notifications from it. So as we all know that fatty acids are the amphipathic compounds which contain long hydrocarbon chain and there is a presence of carboxyl group at the end. And these fatty acids are stored in the adipose tissue in the form of triacylglycerol, which is also called as a neutral fat or triglyceride. It is called as a neutral fat because it doesn't have any positive or negative charge. And the fatty acid mobilized from the triacylglycerol are oxidized to meet the energy needs of the cell. Now for the utilization of stored fatty acids in the form of triacylglycerol in the adipose tissue, it requires three stages of processing. The first stage is lipolysis, which is mobilization of lipid. In this process, triacylglycerol of adipose tissue degrade to the fatty acid and glycerol, and then it is transported to the energy requiring tissues. The glycerol part, which is produced at the end of the lipolysis, it enters into the liver and it gets metabolized over there. So this is the first stage for the oxidation of fatty acid, that is mobilization of lipid. Second stage is the activation of fatty acid. Now the fatty acid which is released at the end of the hydrolysis, it gets activated into the fatty acyl-CoA and then it gets transported into the mitochondria for the further degradation. The third stage is oxidation of fatty acyl-CoA. Over here, fatty acyl-CoA is degraded by the oxidation into acetyl-CoA, and this acetyl-CoA enters into the TCA cycle, which is also called as a citric acid cycle for the complete oxidation. These are the three main stages for the oxidation of fatty acid. So first stage is mobilization of fatty acid from the triacylglycerol lipolysis. So let's see in the detail. So the triacylglycerol, which is stored in the adipose tissue, under the influence of hormone-sensitive lipase enzyme, which gets activated by glucagon and epinephrine, is converted into the glycerol and into the fatty acid. Now this glycerol cannot be metabolized by the adipocytes because they don't have a glycerol kinase enzyme. So this glycerol, it will get entry into the liver via blood and it gets converted to the pyruvate with the help of glycolysis, or it can convert it to the glucose with the help of gluconeogenesis depending on the demand of the cell. Now the fatty acid part, which gets synthesized at the end of the hydrolysis of triacylglycerol or lipolysis process, it enters into the various tissues and it undergoes fatty acid oxidation. At the end of the fatty acid oxidation, there is a synthesis of acetyl-CoA. This acetyl-CoA enters into the TCA cycle and it gets completely oxidized into the carbon dioxide. This is the first step of the oxidation of fatty acid, that is lipolysis. Now we will see how glycerol is metabolized in the liver with the help of glycerol kinase enzyme. So in the liver, with the help of glycerol kinase enzyme, glycerol is phosphorylated into the glyceraldehyde 3-phosphate, and over here there is a hydrolysis of ATP into the ADP. Now this glyceraldehyde 3-phosphate, it gets oxidized into the dihydroxyacetone phosphate with the help of glycerol phosphate dehydrogenase enzyme. Now this dihydroxyacetone phosphate can isomerize into the glyceraldehyde 3-phosphate. If this glyceraldehyde 3-phosphate undergoes into the glycolytic pathway, then it gets converted to the pyruvate. If it undergoes into the gluconeogenesis pathway, that is synthesis of glucose from the various non-carbohydrate resources, then it gets converted into the glucose. So this is the metabolism of glycerol. Now, activation and transport of fatty acid into the mitochondria. So the fatty acid which is synthesized at the end of the lipolysis process is at the outer mitochondrial membrane. So this fatty acid has to be activated into the acyl-CoA with the help of acyl-CoA synthetase enzyme, and over here ATP is converted into the AMP. So over here there is a utilization of two high energy phosphate bond. This activation is essential for the formation of thioester linkage into the acyl-CoA. This thioester linkage is formed between the carboxyl group of fatty acid and thiol group of coenzyme A. So with the combination of this carboxyl group and with the help of this thiol group of coenzyme A, there is a formation of thioester bond in the acyl-CoA, and this fatty acid gets activated into the activated fatty acid, that is acyl-CoA, and the enzyme is acyl-CoA synthetase. Now this activation is occurring in the outer mitochondrial membrane, and oxidation of fatty acid is occurring inside the mitochondria. So this activated fatty acid has to be transported from the outer mitochondrial membrane to the mitochondria. And that transport is occurring with the help of one transport system which is called as a carnitine transport system. This carnitine gets synthesized from the two amino acid, that is lysine and methionine. With the help of this lysine and methionine, there is a synthesis of this carnitine, and carnitine helps in the transfer of this activated fatty acyl-CoA from outer mitochondrial membrane to the mitochondria. So that is the carnitine, which is also called as a beta-hydroxy gamma trimethyl ammonium butyrate, which is synthesized with the help of lysine and methionine amino acid in the liver and in the kidney. This is the image showing the transport of activated fatty acids from the outer mitochondrial membrane to the inside of mitochondria. So in the cytosolic side of the mitochondria, acyl group from the acyl-CoA is transferred to the hydroxyl group of carnitine, and this carnitine gets converted into the acyl carnitine with the help of enzyme which is called as a CAT1, that is carnitine acyl transferase 1. And this acyl carnitine, which is present in the cytosolic side, with the help of translocase enzyme gets transported inside the mitochondria. And acyl carnitine, again acyl group from this acyl carnitine gets transferred to the carnitine. So this acyl group from the acyl carnitine, it gets transferred to the coenzyme A. So this coenzyme A gets converted to the acyl-CoA, and this acyl-CoA will be utilized for the beta oxidation of fatty acid, and this acyl carnitine gets converted into the carnitine. And the enzyme for this step is the carnitine acyl transferase 2. Now the carnitine, which is synthesized inside the mitochondria, is again transported to the cytosolic side with the help of translocase enzyme. So this is how transport of activated fatty acyl-CoA occur inside the mitochondria. This carnitine acyl transferase 1, it is the rate limiting step for the beta oxidation of fatty acid. Now the fatty acid which contains 14 or more carbons require this transport system because they cannot transport inside the mitochondria without the help of this carnitine transport system. And if there is any problem with this carnitine acyl transferase 1, or if there is any problem with this translocase enzyme, or if there is any problem with the synthesis of carnitine, then it leads to the exercise induced muscle cramps, and in the serious cases it can also lead to the death. Now, beta oxidation of saturated even carbon fatty acids. So the beta oxidation, there is a two carbon units are sequentially removed beginning from the carboxyl end of the fatty acid in the form of acetyl-CoA, and it is called as a beta oxidation because oxidation of fatty acid occurs at the beta carbon atom, and this pathway occurs in the mitochondria. So this is the picture showing the structure of palmitic acid. Palmitic acid contains 16 carbon. First carbon, which contains carboxyl group, is numbered as a first carbon. Then second carbon number, it is also called as the alpha carbon, and the third carbon, it is also called as a beta carbon. So in the beta oxidation of fatty acid, there is a oxidation occurs in between the alpha carbon and beta carbon, and the two carbon units are sequentially removed from over here in the form of acetyl-CoA. And the last carbon over where which methyl group is attached, it is called as a omega carbon. So this is the structure of palmitic acid. Now, sequence of reactions of beta oxidation. So there are the four main reactions which occurs in the beta oxidation. First reaction is catalyzed by the oxidation by the FAD. Second step is the hydration step. Third step is the oxidation by NAD, and the fourth step is the cleavage or thiolysis. Thiolysis means breaking of the thioester bond that we have synthesized in the activation of fatty acid. So this particular step which we have already discussed, that fatty acid gets converted into the activated fatty acid, then this activated fatty acid is transported inside the mitochondria with the help of carnitine transporter, which synthesized from the lysine and methionine amino acid. Now, fatty acyl-CoA, it undergoes FAD-linked oxidation, and the enzyme is acyl-CoA dehydrogenase. So at the end of this FAD-linked oxidation, FAD gets converted to the FADH2, and this fatty acyl-CoA gets converted to the alpha-beta unsaturated fatty acyl-CoA, which is also called as a trans-acyl-CoA. It is unsaturated fatty acid because there is a formation of double bond between the alpha and beta. So after the first step, this alpha beta unsaturated fatty acyl-CoA undergoes the hydration reaction with the help of enoyl-CoA hydratase enzyme, and there is an addition of hydroxyl group at the beta carbon. So this alpha beta unsaturated fatty acyl-CoA gets converted to the L-beta hydroxy fatty acyl-CoA because there is a addition of hydroxyl group in the beta carbon. That's why it is called as a beta hydroxy fatty acyl-CoA. Now this beta hydroxy fatty acyl-CoA undergoes NAD linked oxidation. So with the help of NAD linked oxidation, with the help of enzyme beta hydroxy acyl-CoA dehydrogenase, that beta hydroxy fatty acyl-CoA gets converted to the beta keto fatty acyl-CoA because there is a formation of keto group, there is attachment of keto group in the beta carbon. So that's why there is a synthesis of beta keto fatty acyl-CoA, and the NADH, which is synthesized at the end of this oxidation step, it enters into the electron transport chain. Now this beta keto fatty acyl-CoA, with the help of thiolase enzyme, it gets converted into the acetyl-CoA, and in the fatty acyl-CoA, which is shortened by two carbon atoms because two carbon atoms are removed in the form of acetyl-CoA, and this acetyl-CoA will enter into the citric acid cycle. Now for the oxidation of 16 carbon containing palmitic acid, we require total 7 cycles of beta oxidation with successive removal of 2 carbon units. So at the end of this total 7 cycle, there is a synthesis of 7 molecules of NADH, 7 molecules of FADH2, and 8 molecules of acetyl-CoA. So this 8 molecules of acetyl-CoA will enter into the TCA cycle. Now, energy yield from the beta oxidation of fatty acid. This is a very important topic because it is frequently asked in a various multiple choice question, as well as it is also asked in a Viva question. So, complete beta oxidation of palmitic acid, which contains 16 carbon, occurs through the 7 cycles of beta oxidation, yielding 8 molecules of acetyl-CoA, 7 molecules of FADH2, and 7 molecules of NADH. So, number of ATPs formed from the 7 molecules of FADH2 is 10.5 ATP because 1 molecule of FADH2 gives rise to the 1.5 molecules of ATP. Then there is a synthesis of 7 molecules of NADH. So at the end of the 7 molecules of NADH, there will be the synthesis of 17.5 ATP because 1 molecule of NADH will give rise to the 2.5 molecules of ATP. And the 8 molecules of acetyl-CoA, which will enter into the TCA cycle, it will give rise to the 80 molecules of ATP. So there is a synthesis of total 108 ATPs. Now, there is also utilization of two high energy phosphate bonds for the activation of fatty acids. So, the total amount of ATP synthesized at the end of complete oxidation of palmitic acid is 106 ATP. So, 106 ATPs are synthesized at the end of the beta oxidation of fatty acid. Now, regulation of beta oxidation. So, as we have already seen, the main rate limiting enzyme for the regulation of beta oxidation is catalyzed by the carnitine acid transferase 1, that is called as a CAT1. And so CAT1 is an allosteric enzyme and it gets inhibited with the help of malonyl-CoA. Malonyl-CoA gets synthesized in the de novo synthesis of fatty acid with the help of acetyl-CoA carboxylase enzyme. So in the well-fed state, due to increased level of insulin, concentration of malonyl-CoA will increase because insulin will stimulate the acetyl-CoA carboxylase enzyme. So it will inhibit the CAT1. It leads to decrease in the fatty acid oxidation. While in the starvation, when there is an increased level of glucagon, concentration of malonyl-CoA will decrease. So there will be the stimulation of fatty acid oxidation because CAT1 will be stimulated. This is the picture showing the two scenario, that is the well-fed state and in the starvation state. So in the starvation, glucagon, there is a high amount of glucagon. So this glucagon will inhibit the acetyl-CoA carboxylase enzyme. So there is a reduced synthesis of malonyl-CoA. So there will be the stimulatory effect on the CAT1, that is carnitine acid transferase 1. While in the well-fed state, there is a increased amount of insulin. So it will be having stimulatory effect on the acetyl-CoA carboxylase, so it will lead to increased synthesis of malonyl-CoA. So this malonyl-CoA has a inhibitory effect on the CAT1, so there is an inhibition of beta oxidation of fatty acids. Now, defects in the beta oxidation. So the abnormalities in the transport of fatty acid into the mitochondria and defects in the oxidation can lead to the deficient energy production by the oxidation of long chain fatty acids, and it can lead to the hypoketotic hypoglycemia, hyperammonia, skeletal muscle weakness and liver diseases. Acyl carnitine accumulates when there is any defect in the transferase or translocase enzyme is there, and dietary supplementation of carnitine has been found to improve the symptoms in some cases. So this is about the defects in the beta oxidation. Another is the Jamaican vomiting sickness. This Jamaican vomiting sickness is occurred due to the consumption of ackee fruit that grows in the Jamaica, which contain toxin named hypoglycin. This hypoglycin, it inhibits the acyl-CoA dehydrogenase enzyme, the first enzyme which is required for the beta oxidation of fatty acids. So during the consumption of this ackee fruit, it leads to the hypoglycemia, sudden onset of vomiting, convulsions, coma and death. So that is regarding the Jamaican vomiting sickness. Now, oxidation of fatty acid with the odd number of carbon atom. We have seen the oxidation of fatty acid with the even number of carbon atom. The oxidation of fatty acid with the odd number of carbon atom, it is same as that of beta oxidation of fatty acid. So there will be the removal of acetyl-CoA from the odd number of carbon, odd number containing fatty acid until 3 carbon propenyl-CoA residue remains. And so there will be the removal of acetyl-CoA through the beta oxidation of fatty acid from this odd number carbon atoms until 3 carbon propenyl-CoA residue remains. So at the end of the oxidation of fatty acid with the odd number of carbon atom, we are having propenyl-CoA as well as acetyl-CoA. This propenyl-CoA, which is synthesized, it will be metabolized with the help of vitamin B12 and it gets converted to the succinyl-CoA, which is a constituent of citric acid cycle. So this is how this propenyl-CoA, which is synthesized at the end of the odd chain fatty acid, converted into the succinyl-CoA. First it gets carboxylated with the help of biotin into the d-methylmalonyl-CoA. This is the carboxylation reaction. So there is a addition of carbon dioxide and there is a utilization of ATP. So this d-methylmalonyl-CoA, it gets converted into the L-methylmalonyl-CoA with the help of epimerase or racemase enzyme, And this L-methylmalonyl-CoA, with the help of methylmalonyl-CoA mutase enzyme, it gets converted to the succinyl-CoA. And over here we require vitamin B12 as a coenzyme. So if there is any deficiency of vitamin B12, then there is no conversion of L-methylmalonyl-CoA to the succinyl-CoA occurs. So there will be the accumulation of methylmalonyl-CoA in the blood as well as in the urine, and there will be the excretion of methylmalonyl-CoA in the urine occurs. This is how propenyl-CoA gets converted to the succinyl-CoA, and this succinyl-CoA is the constituent of PCA cycle. Now let's see the various multiple choice question. Number of ATPs found by the oxidation of one molecule of palmitic acid 16 carbon, which was asked in Kerala 2009, and the options are 146, 106, 135 and 34. As we all know the answer that after the oxidation of palmitic acid, which contains 16 carbon, there is a synthesis of 106 molecules of ATP. So the correct answer is B. Third question, which was asked in AIIMS November 2011. In the well-fed state, activity of CAT1, that is carnitine acyltransferase 1 in outer mitochondrial membrane, is inhibited by. And the options are A. Glucose, B. Acetyl-CoA, C. Malonyl-CoA, and D. Pyruvate. So the correct answer is Malonyl-CoA. In the well-fed state, there is an increased availability of insulin. So there is a stimulation of acetyl-CoA carboxylase enzyme. So there is an increased availability of Malonyl-CoA, and this Malonyl-CoA is the inhibitor of CAT1, that is carnitine acyltransferase 1. Opposite occur in the starvation. So the correct answer is C. Malonyl-CoA. And the third question, which was asked in the JIPMER 2013, one of the following is obtained in the beta oxidation of odd chain fatty acid, and the options are A. Acetyl-CoA, acetyl-CoA, B. Acetyl-CoA plus propenyl-CoA, C. Propenyl-CoA, propenyl-CoA, and D. Acetyl-CoA. So as we already discussed, at the oxidation of odd chain fatty acid, there is a synthesis of acetyl-CoA plus propenyl-CoA, and this propenyl-CoA gets converted to the succinyl-CoA with the help of vitamin B12. So the correct answer is B. Acetyl-CoA and propenyl-CoA. These are my references. Thank you for watching. Please like, share and subscribe Biochemistry Basics by Dr. Amit. And don't forget to press the bell icon so you can get all the notification from it. Thank you.